Good morning, everybody. Today, after the Mendel, some other biologists studied in the genetics and found out some deviation to Mendelism. Now, today I want to teach you about the deviation of Mendelism. There is how incomplete dominance, co-dominance, etc. Already I stated about the incomplete dominance in the tobacco plant prior to Mendelism that J. Collicutor performed the studied in the height of the tobacco plant where he saw the incomplete dominance, that is the in the F1 generation the offsprings are intermediate in height, not tall or dwarf. Now the incomplete dominance gradually seen in the plant that is called as 4 o'clock plant, where there is two types of flowers are produced. One is white and another is pink. Now the there the um, red and white. There the red is the dominant form and the white is the recessive form. When the red and white are crossed with each other to form the pink type of plant. Red crossed with white in the F1 generation, that is pink plant is you see here the one is red, that is capital R smaller, and one another is white, and in the F1 generation they produced pink plant. And so that there is incomplete dominance, there is no complete dominance, not the red plant is produced, which is called as incomplete dominance. And also for the incomplete dominance we can see other example like that tobacco plant, etc. And another plant also snapdragon where there is incomplete dominance, etc. Then you see here the RR is the red flower bearing plant, and smaller is the white flower bearing plant. In the F1 generation, that is production of the pink plant. Then when the F1 generation capital R smaller crossed with each other, in the F2 generation the results, these are shown as found. The red one is red and another two, this is two are pink and another one is white. Now this is the table of the F2 generation, and the ratio is the Mendel's ratio, monohybrid cross ratio or monohybrid cross ratio, which is the 3:1 phenotypic ratio, but that is genotypically 1:2:1, that is deviated, that is transferred to 1:2:1. Then next another the BF co-dominance. Co-dominance means how the other form of genes are also given importance, that is the co-dominance. Co-dominance generally is the bright example in the case of the human beings. Human beings consisting of one gene, that is I, which produce the antigen in the red blood cell according to surface of the red blood cell, according to the type of antigen to be produced, the red blood cell as called as it, and I is the gene which is responsible for the production of the antigen. Now if there is suppose dominant allele form of this gene, like capital I and capital A produce antigen A, and the blood group of that person is A, and another dominant allele also of the gene I, that is capital I capital B, that is produced antigen B, which gives rise to blood group B, and when the F1 generation of the capital I capital A and capital I capital B married together, the F1 generation which found having the capital I capital B and A and capital I capital B forming the F1 generation, the progeny having AB blood group. Here you see the progeny having AB blood group here. Here are the P1 parent capital I capital A A and produced capital IA product, and here capital I capital B capital A capital B produced gamete produce capital IA gamete, and here also capital I capital B gamete. Then in the F1 generation capital IA capital IB produced, and that is having AB blood group. That is both the dominance character cooperate with each other, cooperating with each other produce the AB blood group possessing plant. Possessing human being. Sorry, this is seen in the human being. Then in the F2 generation, when these F1 progenies are intercrossed with each other, produced two type produced two type gamete, male produced two types gamete, capital I capital IA and capital IB. Similarly, the female also produced two types of gamete. The parent one produced two types of gamete, capital IA and capital IB. And then these four types of progenies which are produced, among which 1/4 progeny have blood group A, other 1/4 progeny having blood group B, and half progeny having blood group AB. In this manner, the experiment is same, that is the ratio is again diverted to 1:2:1 due to this codominance also. For incomplete dominance the ratio is diverted, and for codominance also the ratio is diverted. That is changed to 1:2:1. So this is about the incomplete dominance and codominance. Today, thank you. I end with this, and I myself, Dr. Patya Das, as I told you, please follow my channel for your study, and like it, share it, and subscribe it. Thank you all.